Hi everyone, welcome to the very first lecture for this course. Right now in the class you should be working on assignment 0, getting that finished up for the due date that's upcoming. Remember, that's just a course on academic honesty. You take the course, you get your certificate, and then you upload that. It is required for you to complete that assignment to pass the class, so if you don't complete the assignment, no matter what your other grades are in this course, you can get at most a 49% in the class, and you would get a comment of INF or incomplete fail on your transcript. So if you have any questions about that, please reach out. It really should only take about half an hour-ish. I think that's what it took me when I went through it. And it's, if you have a certificate that's within a year old, you can use that as well and it would still be valid. I realized in my intro video I forgot to introduce my virtual teaching assistants. I have, let me get my pointing right. This one here is Rambo, and this one here is Vader. Those are my real life dogs. They are Frenchies though, so I can't have them in the room with me when I record these videos because they snort and snore and it gets picked up by the mic and is very distracting. So we will just deal with these cute characters instead. I am really excited to get into the course content. This very first topic is my most favorite topic. It is so near and dear to my soul. As I might have mentioned, my undergrad was in biochemistry. I'm a big biochem nerd. It's always going to hold a special place in my heart, and so I'm excited to go through some of those basics with you. Just a couple housekeeping items before we get into the content. These first few lectures in this class are going to be more traditional lectures where we're going through slides and I'm just adding some extra color, some extra explanation on top of it. Once we get into more of the programming that we're going to be doing in this class, it's more flipped where we'll be doing exercises and be building up some muscle memory of actually coding and going through some together, and then you'll be expected to do some on your own. These initial lectures that are going to be going over like the biochemistry, some intro bioinformatics things, they are not going to be as long as if we were in person because we don't have that back and forth going on that we would have in a traditional lecture where I would be asking questions, waiting for answers, maybe clarifying things, etc., etc. But so the videos will be shorter, but I still would love to have a little bit of that back and forth happening on the discussion boards. So I will be asking questions. I do have the expectation that you will engage in the discussion boards to answer some of those questions so that I can get a good sense of what is sticking well and what maybe needs some more explanation for the class. This will help us make sure that we're all on the right track when it comes to things like our midterm and our final exams and even our assignments and things like that. All right, so let's jump into it. We are going to just be going over a very, very small portion of biochem. You have taken the prerequisites for this course, so this should all be review at this point. It will be a good gut check as we kind of go through these slides, like, hey, do I remember this? Do I need to maybe ask him some clarifying questions? Maybe go and find some more resources on X, Y, or Z topic just to make sure that I really understand what this is doing. We're just going to start off with a small section of it that we need for the initial kind of bioinformatics things that we'll be learning. And then as we ramp up with more advanced bioinformatics things, we'll be peppering in some different kinds of biochem that are relevant for those. So this is just really going to go over like cells, DNA, RNA, proteins, and the central dogma, that kind of thing. So if you feel really comfortable with this, you might be able to like zoom it up to 2x speed and get through it really quickly. But if you would love the refresher, that's what I'm here for. And I am so passionate about this information, so I might actually talk a little bit too fast. But the good thing is we're on YouTube, so you can slow me down if I get too excited talking about my favorite things. So we're going to start off with a little bit about cells and cell theory. So in my brain, when I think about a cell, I still go back to thinking about a Lego, a piece of Lego. We can take that piece of Lego and add other pieces of Lego to it and build all sorts of things. We can build a house, we could build a car, but at the end of the day, we still have that individual piece of Lego that we're using to build up all of these other things. The same idea with cells. So with a cell, you can have an individual cell that makes up a whole organism in the case of something like a bacteria, but you can also build up with multiple cells to have these more complicated organisms, things like humans, things like plants, dogs, my pointing, all sorts of good things like that. Every single cell is going to share the same kind of basic chemical composition. They're going to contain things like genetic information that can be passed on from one generation to another generation. They're going to contain things like proteins that are going to help them do the various tasks that they need to do with that genetic information. All cells that we have exist or originate from pre-existing cells. So we'll talk a little bit about evolution. We'll talk more about evolution later on in the course, but that idea around, hey, this thing didn't just come out of thin air, it came from something that was very similar to it before, is very, very important, and you'll see why later on in the course when we start talking about phylogenetics and things like that. There are two main types of living organisms that I've already kind of touched on briefly. Pop quiz, don't look at the slide, but you probably already did. What are the two types of living organisms? We have prokaryotes, which are unicellular, so those are going to be single cell organisms. These cells lack a nucleus, which is like the home of all that genetic information that's bound by a membrane of lipids or fat. And you'll typically see a prokaryote in things like bacteria. We also have eukaryotes, which can be unicellular, so single cell, but are more typically what we would think of as multicellular, so things like humans and plants and stuff like that. The main difference between those two types of cells is not the number of cells that is needed to build those different kinds of organisms, but it is truly just that nucleus, that membrane-bound subcellular structure that would contain things like our genetic information. So eukaryotes have a nucleus, prokaryotes don't have a nucleus. So this gives you a little bit of a visualization here on what I'm talking about. So if we take a look at this and I was to say, which one of these is the prokaryote and which one is the eukaryote? Would you be able to answer that? I think I can write on this. Let me just look at this. Here we go. Yeah, draw. So over here, this is our prokaryote. I'll put a P on it just so that we're labeling it here. You can see, maybe let me pick a different color maybe than red. That's kind of hard to see on there. Oh, I probably shouldn't pick blue either because, no, blue is fine. I'm trying to remember for colorblind if blue is differentiating from red. And I apologize for anyone who is colorblind if my colors that I've selected here aren't visible. Please just reach out. I can find you a colorblind friendly copy. But you'll see in this prokaryotic cell, we have no nucleus. So we have all of this genetic information in here that's kind of collected together inside of the cell, but it's not bound by anything. It's just same is attracted to same in the cell. It's sitting there together in a similar spot, but there's nothing holding it there. It could move around. It could be more dispersed throughout the cell. Whereas with our eukaryotes, which we have over here, you'll see we have a nucleus, which is this part right here. It's kind of actually very hard to see for my eyes in this image, but I'll share some other ones on Canvas. And what this is it's actually a three-dimensional sphere type shape-ish, loosely speaking, where it has that lipid membrane with some protein pores so things can pass in and out of it. But all of that genetic information is typically contained within that nucleus. So it's right there. Oftentimes it's drawn in the center of the cell, but that doesn't mean it only exists in the center of the cell. It just means that it has that lipid membrane that's containing all of that information. Whoops. One thing that we haven't talked about yet is viruses. So we've talked about things like people and plants and bacteria, but not viruses. Viruses are a little bit different. They are not considered living organisms. Bacteria are considered living organisms. The reason they aren't is because they require a host. So you can think of this as like a human or an animal or a plant, anything that they would infect. They require that host's cellular machinery to replicate their genetic information. And so this is either can be DNA or RNA, and it's going to be wrapped inside this protein coat. So it's really lacking some of the machinery that we would consider it would need to be living. So if a virus was just floating out there on its own, it wouldn't be able to make a copy of itself without that additional organism that it can infect and leverage that machinery for. When we're thinking about how do we make these new cells, what is actually happening here, it all comes back to the central dogma of biology, which I'm sure you have heard time and time again, but it's such a very important and fundamental dogma. Dogma? It is all about how information flows and is replicated in the cell. So typically what we'll see with the central dogma is we'll have our DNA molecules, which is our genetic information that gets passed on generation to generation. It will get converted into RNA, either like think of messenger RNA, that type of thing, which will then get converted into a protein or like the thing that we actually need more of in our cell. So the DNA is like our instructions. The RNA is us taking a piece of those instructions and saying, hey, we need to build a bookcase today, for sake of argument. And then the protein would be the bookcase itself. So it's kind of nice to sometimes think about it as like, pretend you had all of IKEA's instruction manuals. It's just available to you and all of the building blocks you needed. So if you needed a piece of furniture from IKEA, you just had to go into your file system, find the instruction manual related to the piece of furniture you wanted. So the instruction manuals would be our DNA. Once you have that instruction manual, you would then take that and combine, and that would be your RNA. So that's your instructions for how you're going to make this specific thing. So usually RNA is related to like a specific gene that we're talking about, that kind of thing. So what specific thing from IKEA are we building? And then we go in and we start doing the process of, okay, let me get that piece and the screw over here and put these things together. And when we end up with our final three-dimensional product that we wanted to build, say it was a bookshelf, say it was a chair, whatever it was, that would be representing our protein. So we have a couple of different processes in there, and it's kind of, maybe my analogy is a bit hard to follow, but your DNA is going to be your collection of your instructions. Finding the right instruction in the DNA and pulling it out is going to be the process of transcription to generating the RNA, which would be the individual instruction book, and then taking that individual instruction book, combining it with those components you need to build the final product that you're looking for is translation, and then that end result would be your protein. So if that was a useful analogy, great. If it wasn't, throw it out. Don't worry about it. Sometimes I found when I was taking these classes, I was so deep into it that I was like, I don't need these analogies. I just need to think about all these wonderful little molecules that are floating around, and that's great. But I do find, since we can't actually see them, that having those analogies sometimes can be helpful. It is also possible for the central dogma to only have RNA and have that go back into DNA. This happens more commonly in things like bacteria and those kind of prokaryotic cells, but it is possible. One thing that is not possible is for a piece of protein to go back into RNA or for a piece of protein to go back into DNA. So once it reaches this protein state, it is kind of like an end state. It can't be broken back down into RNA or DNA or red reverse. So it always has to flow this way. We can get a little bit of this going on where the RNA goes back into DNA. But once we hit the protein, that's the last stop of the train station. That's what we're going to be working with. It cannot go backwards to RNA or DNA. So here's a little visualization that can help if my analogy did not help. So up here we have our DNA. So this is just a chunk of DNA. Typically in an organism, we'd have a lot more DNA, and we're going to see examples of that once we get into looking at some of the data. But so this is just a representation here of an individual gene where we have our promoter region, and then we have some exons, which are these red sections here, and some introns, which are these black sections here. So this piece of DNA will get transcribed into mRNA and then transcribed and processed into mRNA. The processing part is where these introns here are removed or spliced out. And then we also get things like the 5' cap added and the 3' poly A tail. Once we have that, it goes through a process of translation where that mRNA is converted into a protein or a peptide strand. So each of these little different circles that we have up here represent a different amino acid that gets coded based on our codons. So we're going to go through all of that in a little bit more detail, but that kind of gives you a high level overview. Once we have that peptide chain, it's going to start folding up on itself and making this three-dimensional structure. There might be some post-translational modifications that happen, like phosphorylation, to give us our active protein at the end. So DNA is my favorite part of biochem. I'm so biased because I love it so much. I did my master's and PhD in three-dimensional genome structures. So it's my favorite. I'm biased. I hate to break it to you. Feel free to let me know in the discussion board what your favorite biomolecule is, whether it's something like DNA or protein or RNA. What's your favorite? I would love to know. So DNA is what holds all of our genetic information that's passed through inheritance. So it holds all the instructions our cells need to help us live and to help other organisms live. I find that, and I will apologize, when I'm explaining this often, I talk about it in the human sense because I feel like that's the most tangible. But it is really important to remember that all of these things are valid for like plants and bacteria, etc., etc. Okay? It is in eukaryotes, we typically have our DNA stored into these things called chromosomes, which are just individual structures, individual units of information, and then they're going to be stored within that lipid substructure in the cell called the nucleus. There's also some DNA that gets stored in other organelles like mitochondria, which is the powerhouse of the cell. And that, if you are remembering from your previous classes, is DNA that also will get passed down generation through generation, but through a maternal line. So DNA has a very defined structure. It's a macromolecule that has a base here, which we would call a nucleoside, that's going to have 5 carbons on it. On that base, we also get on the 5' carbon this phosphorylated tail, and then on the 3' carbon, this oxygenated tail. These things here are what gives DNA the directionality that we'll talk about later. Onto this, you can get additional things like or additional types of DNA. So we can have purines, which would be our adenine, which we often represent with an A in bioinformatics, or guanine, which we often represent with G, or our pyrimidines, which are cytosine, which we represent with a C, thiamine, which we represent with a T. This picture also has uracil in here, but that's an RNA thing. That's not a DNA thing. We represent that with a U often in bioinformatics. So we have different ways that we can represent this. The biggest thing that I want you to remember is that it has these directionalities associated with the 5' and the 3' end. These different bases here, which so our purines or our pyrimidines, would get attached to this carbon right here. And so there's all these complicated structures that are happening at the molecule level that are going to lead to different kinds of structures that we might have to deal with in some of our bioinformatics tools that we learn about. So what ends up happening is those individual bases. So remember, we have our 5 carbon ring. We have our base over here. That's going to be our A or T or C or our G. And then we have our phosphate over here. These are going to be combined with other versions of this. So they could also have guanines, they could have thiamines, whatever, into forming these really, really long chains. So they come up and they form this chain of these sugar phosphate backbones. So the linking happens through this phosphate tail that we see here, and it will attach to the 3' OH on the previous base's structure. So because we're always adding to the 3' OH when we're building up these molecules in our cells, that's why we have directionality. The beginning of it will always be the 5' end, and then the end of it is always going to be the 3' end. So when we have a single strand of DNA. Usually when we're writing a DNA sequence in bioinformatics using like those letters that I was talking about, those A's, T's, C's, and G's, we are conventionally writing it from the 5' to the 3' end. So that directionality is really important, because you'll see later as we get into some of our bioinformatics tools that sometimes when we're taking that sequence and then we're translating it into, say, a protein sequence, if it isn't in the 5' to 3' orientation, we would get completely different results. And it would be completely misleading as to what's actually going on with that biomolecule. So always, always in this class, assume that we are writing things from the 5' to the 3' end, unless we actually specify when we are writing something that we have it at the 3' end on one side and the 5' end on the other side. So this would mean that if we were trying to convert to RNA, we'd actually have to start looking at the 5' end and then going from that side. Typically, DNA doesn't exist as a single strand. There's lots of great biochemical reasons for that. Typically what we have is a double-stranded piece of DNA. And that's when we get that wonderful helix that we often, wow, the drawing that we see so often as represented from DNA. This is why I'm not an artist. But we often get, this double-stranded piece. So we'll have two different strands of DNA. Let me pick another color for one of my strands. Wow, that was a bad color choice, but that's okay. It's fine. Everything is fine. And one of those strands is going to run from 5' to 3', and the second strand will be complementary, so it's going to run from 3' to 5'. So they're always kind of matching up. And if you remember from the prerequisites to this course, we always typically see G's matched with C's, and I think we have some over here, A's matched with T's. So that's kind of the typical base pairing that we would be seeing. How they're matched up is through hydrogen bonds. So we're not going to get into that too much in this class. There's other courses for that, but that's how that structure is being held together. When we want to make RNA out of DNA, oh goodness, I'm getting ahead of myself. When we want to replicate our DNA, so make another copy of it, say our cell is dividing, we want to make a new cell. We need to make a copy of that in order to give it to the new cell. That would be like saying, in my IKEA analogy, we're going to make a new IKEA store where you can buy these collections of instructions, the building blocks and etc. We would have to go through and photocopy all of those instructions to give them to this new store. Same thing happens with our cells. When a new cell is being made, it needs copies of all of those instructions so that that can be given to that new cell so that it has that to work from. This is called replication. Replication will happen on both strands of our DNA. One will be called the leading strand, so where you have one will be called the leading strand and one will be called the lagging strand. And so the lagging strand is when the polymerase that is opening up this section here, it has to go in this direction. So it will have to come back around once more is opened up and do more and then link it all together. The leading strand, it can just run with the polymerase and it just keeps on going this way. I'll try to find a video. There used to be this really excellent video demonstrating what this actually could look like in the cell. And it shows like how that polymerase goes and then how these little, the strands break and then come back together at the end of the bubble and build up these new DNA fragments, etc., etc. So if I don't find that video, bug me in the discussions, but I should be able to find that for you. DNA replication is typically very, very accurate, but occasionally mistakes do happen. That's okay. We all make mistakes. Even our DNA makes mistakes. So there's lots of different processes that the cells have built into themselves for correcting these errors. So they could be things like when they go through and they start proofreading it, they see that there's the wrong nucleotide was added, it will actually cut that out and then try to add the new one in. But still, mistakes persist even with all these checks and balances that they have in place. And that's when we get things like mutations that come up in our DNA. And so those mutations are really important in the bioinformatics space, because oftentimes what we're looking for is we've sequenced up some sort of genome or some gene or whatever that might be, and we want to identify why this specific organism or group of organisms has these different characteristics when compared to the reference. So we want to find where those mutations are and what could be happening to cause some sort of what we would say phenotype that we're seeing. So I remember back when I was working at Ag Canada, we were working on trying to identify why certain types of mustard plants had different kinds of oil compositions in them. And so we could then identify which ones were related to the oil compositions we wanted and breed selectively for that. So we were sequencing the genes associated with the producing those different kinds of oils and then comparing them to each other to figure out, okay, where are the differences in this DNA here? Why is this one having more of this type versus this other type? And that was what I did for a research project one summer. It was really, really fun. Mutations are also really important when it comes to things like evolution. We wouldn't exist if it wasn't for mutations, likely. I mean, that's probably a bold statement for me to make, especially if I'm putting this on YouTube. Let me walk that back a bit. It's possible we wouldn't exist if it wasn't for mutations. Maybe there'd be some other way that we got here. I'm not sure, but mutations are what has made all of these different organisms that we have today. So if we look back, once we get to phylogenetics, you'll see we can identify common ancestors by looking at the DNA and the mutations that individuals have, whether that's in plants, in people, in dogs, and you name it. And then we can see what's more related to other things and identify things like paternal and maternal relationships. We can also identify, if we're looking at bacteria, if we have a new bacteria come in and we're like, hey, this gene or genome is really similar to this other one that we've seen before, maybe we can utilize some of the drugs that we used for that bacteria to help treat this one. So there's lots of different cool things we can do with mutations. Genes, I've mentioned them a bunch of times now. But a gene is just a sequence of DNA that codes for a protein. So it is a very defined part of a genome. So genome is going to be the whole collection of DNA, and a gene is just a piece of that. So it'd be like a single instruction booklet for building a very specific type of bookshelf at IKEA. The human genome has 3.2 billion base pairs. I should double check that number to make sure it's still accurate. And there are less than 30,000 genes. So that seems like, oh, okay, I don't know, maybe that's reasonable. But when we actually look at what proportion of the genome is made up by gene coding DNA, it's only 2% of the human genome. So 98% of the human genome doesn't encode for genes, which is wild when you think about it. Why would we be holding on to all of that information if it wasn't necessary for something or wasn't needed, et cetera, et cetera? So that was like the bread and butter of my PhD thesis. I could talk about this for ages, but I will stop myself now. In eukaryotes, genes will contain introns and exons. So exons are actually the coding regions, as I mentioned before. Introns are non-coding regions. So introns get spliced out as a pre-mRNA gets converted into an mRNA. Sorry, my phone is going wild. I apologize about that. So once we see our mRNA, it won't have any of those introns. They will all be removed. But if we were just looking at the DNA and trying to figure out what the RNA would look like, we would also have introns in that as well. So we'll see examples of that when we move on to the bioinformatics tools. RNA is very similar to DNA, but it is single-stranded instead of double-stranded. And instead of using thymine or T as a base, it uses uracil or U to pair with adenine or A. So oftentimes you'll be able to identify when a sequence you're looking at is RNA because it will have a whole bunch of U's in it and absolutely no T's. So that's a dead giveaway that what we're looking at is RNA. There's lots of different kinds of RNA that exist inside of cells. We'll often be talking about messenger RNA, which again, if we go back to the previous slide, is that RNA that's used to code for a protein. But we also have things like transfer RNA, which are RNA molecules that are actually used to build up that protein as it's being read from the messenger RNA, as well as ribosomal RNA. So there's lots of different kinds. Ribosomal RNA is really, really interesting. I won't get into it too much in this class, but I've always found it very fascinating because to me, in my brain, it kind of acts like a protein, but it's not a protein, but it has this very interesting function. So highly recommend that you look up more into that if that also piques your interest. So DNA transcription, what I tried to already talk about earlier when we were talking about replication, is the process of making RNA from DNA. So pop quiz, do you remember what is the central dogma here? What is the order that this has of when we go from DNA into proteins? I want like the Jeopardy music to play here, but it won't. So remember, we have DNA to RNA to proteins, and that is our typical central dogma. So transcription is this arrow here. This very first arrow is the one that we're talking about here. The process of transcription is very, very similar to replication with DNA. So the strand is going to be breaking up into this little bubble where it's going to be reading from each side. No, actually, sorry, not for transcription. Well, it will be reading from one side to build up this mRNA molecule. The main difference between DNA replication and transcription is that there's a different set of enzymes. So the key enzyme here is an RNA polymerase. So transcription is when our RNA polymerase is going to bind to our template strand. So that's the strand that goes from 3' to 5'. And it is going to build up this mRNA molecule as it reads from that. And the reason why it uses this template strand and not the coding strand for this is because it can do the building up of that mRNA molecule in a continuous fashion. So it doesn't have to stop and make breaks and all of that kind of stuff like we would see on the one strand with DNA replication. So the gene is going to be located on the coding strand. So when we're looking at the gene from a bioinformatics perspective, we'll be reading it as the coding strand. And then the template strand here is also sometimes called the antisense strand. We have about 1 million, that's an exaggeration, feels like 1 million names for these different types of strands. Template strand is also called the antisense strand. Coding strand is also called the sense strand. I'm going to try my best to use template strand and coding strand for the duration of this course, just to keep it clear, but just to know that those are important here. The 5' end is usually what we would call upstream from the gene because it's going to the left. And the 3' end is what we would call downstream from the gene because it's going to the right. And when I say left and right, I'm talking about the coding strand. So on the coding strand, and we're going to have things like our promoter where all of these different things or proteins are going to bind. That's going to kick off that whole transcription process. But just remember, and I know this is confusing, I think it took me all 4 years to nail this down, but that the transcription is being read from the template strand to build up an mRNA in the 5' to 3' direction. Here's an example. You're going to have to go to the discussion board if you want the answer to this. We have here our coding strand. And here our template strand. I'm just going to put a T because as we can see, it takes me a minute to write on this thing. If our top strand is the coding strand, which it is, I want you to write in the discussion board what the RNA sequence would be. And then if we use the bottom strand and just gave you that strand, how would you know what the RNA sequence would be? So answer both of those questions. We'll have more discussion about that in the discussion board. Whoops. Oh, okay. Now we're going to move on to proteins. Proteins are my least favorite biomolecule. I think they're still good. They're still good. They're important. They're great. People love proteins. Very important things. I just find them less cool than DNA. I don't know why. I could probably unpack that, but that would end up being a much longer lecture. So proteins, again, are biomolecules that consist of chains of amino acids. We have 20 amino acids. I will never ask you to memorize the amino acids in this course. You will always have like a codon table available to you for any questions that you would have in this course. You don't need to memorize their structures, anything like that. Typically, proteins have 4 levels of structures. Do I have this? No, not on this other strand. So we would have, I think I do actually later on. Here we go. We're going to skip ahead a couple slides so we can talk about this. So proteins have 4 levels of structures. They have the primary structure, which is just if we stretch that chain out end to end, what would that look like? We have secondary structures, which are typically beta sheets and alpha helices. So these are common substructures that we would see in proteins. We have tertiary structures, which are typically like the 3D representation of that peptide folded up onto itself. And then some proteins, not all proteins, have quaternary structures. So a very common example of this is the hemoglobin protein, where multiple tertiary structures come in and interact with each other to give the actual final function. So I'm going to skip back to where we began here. Inside of that primary amino acid structure, so just that chain that we have, remember it's being built up of amino acids. So here is an overview of all the different kinds of amino acids we have with their structures. Occasionally, later on in the course, we'll be talking about how specific amino acids mean certain things when it comes to biochemistry and how we've leveraged that in our bioinformatics algorithms to identify things like when secondary structures are likely occurring given a protein sequence and things like that. But as I mentioned, you will always have this available to you in this course. You do not need to memorize it. So proteins and peptide strands, those primary structures, are very similar to DNA in the sense that they are also directional. So they are going to have an N or an amino end as well as a C or a carboxyl end. And that goes back, so if I scroll back one slide here, whoops, up here, you'll see that we have, let me find a good protein to do here. Actually, I think my other picture might be a little bit easier to explain this. But up here, this structure here that we have is the same on every single amino acid. And it's this part down here that changes. So this carboxyl end is this first C here. And remember with biochemistry and organic chem, we don't have to write the C's. It's just like the lines that tell us where the C's are. So this C here is going to be our carboxyl end, and this C here, or this NH here, is our amino end. And so these are the two sides here that are going to be combined together through a biochemical reaction to build up these chains. It's not anything to do with these bits down here. It's all in this repetitive structure that we see up here. And so when those things are joined, it's called a peptide bond. So you see an example here, we have our COO and our NHH. Those combine together and they make this thing called a peptide bond right here, which has our NH on one side and our COO on the other side, creates a bit of water. That's not important for this course. What is important is that it maintains that directionality, so that N is always going to be on the left side when we think about it in terms of protein sequences, and the C will always be on the right side when we think of our protein sequences. So just like how, yes, we could write 5' to 3' in the 3' to 5' direction, when we are working with protein sequences, we're often writing them in the amino to carboxyl direction. So that's how you're typically going to see that represented. You'll probably also see that represented in all of your biochem textbooks and things like that in that direction. We already talked about protein structure. I'm not going to go over that again, but we will now start talking about protein synthesis. So I actually added a video about protein synthesis onto the course website that you can watch after this that is way better at visualizing and depicting what's going on here than I'm probably going to be able to explain. But this here is a eukaryotic cell. We know that because it has a nucleus. Inside we have our DNA up here that would get transcribed into RNA. I always want to say mRNA, but it's RNA. So remember, our RNA is going to have things like our introns that need to get spliced out. Post-transcription, those will get spliced out, and we'll also have our 5' cap and our poly A tail that's get added to our mRNA. Once that's done, that actually moves outside of the nucleus into the main part of the cell for translation to occur. So that's where all the components for translation actually exist in the cell. Things like our amino acids, things like our proteins that we need for that process, they're in the main part of the cell, not in the nucleus. So once we have that, our ribosomes can start binding to our mRNA, our transfer RNA can come in with those amino acids and start adding it up and building up this polypeptide chain that we see here. Once we have that polypeptide chain, it's going to start folding in and up on itself. It might get some additional biochemical modifications like phosphorylation, and then it's going to become an active protein within our cell. The process where this how this happens is a little bit confusing, because if you just looked at DNA, you wouldn't be able to say, oh yes, this is going to be the structure of this protein, at least not easily. That is a goal that people have in bioinformatics, but we are not there yet. What we need to do often is we need to convert, okay, we take our DNA, we'll convert it to RNA, and then we're going to convert it into a protein. And the way that we do that, because it's not as easy as it is with RNA, where we can just swap out a U for a T, is we actually have to work with those things called codons. So remember, codons are triplets of bases in our DNA and our RNA that represent a specific amino acid being brought into that growing polypeptide chain. So we have a whole bunch of options that we could have for codons, and then we have redundancy within that codon code, so that there's multiple codons that can code for the same amino acid, because we don't have 64 amino acids. Translation will always start at a very special codon called the start codon. Pop quiz, what is the start codon? And ends at a very special codon. There's 3 of them, called the stop codon. You can also put that in the discussion board. I don't think it's on the next slide. It probably is. You should be able to find that information on here. And then you can throw it into the discussion board and I can give you a stamp of approval. So in this course, you'll always have a codon table available to you. We do often need to do that translation from RNA into a protein sequence. And we'll be able to use things like this in order to do that. So here it shows, say if you had 3 U's, then that's going to be replaced by a phenylalanine in the polypeptide chain. Again, if you had ACC, that's going to be replaced by a threonine in the polypeptide chain, and so on and so on. This video doesn't work anymore. Trash it. I posted one for transcription and translation on the site that I mentioned earlier. You can take a look at that and let me know if you have any questions. That was a lot of information. A lot of me talking about how much I love DNA and don't really love proteins, but that's okay. If you have any questions, reach out to me. You can send me an email. You can post in the discussion board. You can come to help hours. I am happy to help. Next chapter, which you're probably going to watch right after this, we're going to be diving into biological data sets. See y'all.